Good morning, student. Today I will give a made easy class for the immunoresponse. This paper that will be shared in WhatsApp, that actually it is taken from the Lippincott, but it is from the old Lippincott. Even if you try to find this immunology map in the recent edition of Lippincott microbiology, you will not get it. So please print out this immunology map, this full paper, and stick it on your wall and please see it regularly and try to understand. And this, I think that it will be very much helpful for understanding, and also not only the microbiology but the immunological concept of medicine also. So please listen. Already you know that in immunology there is two type of immunity, then one is the humoral immunity, another one is a cytotoxic immunity or cell mediated immunity. Okay, now the B cells mediate the humoral immunity. This one, please see the left hand side. B cells mediate the humoral immunity or antibody mediated immunity, and the T cell, they mainly mediate the cell mediated immunity. And these T cell are two type. One is CD4, another one is the CD8, and CD stand for cluster of differentiation. Now you know that CD4 T cell is called the helper T cell, and CD8 T cells, they are also called the cytotoxic T cells. So mainly the cell mediated immunity is mediated by CD8 T cell, that is cytotoxic T cell, and humoral immunity, it is mediated by the lower one, B cells. Okay, just concentrate on the left side of this paper. Now see, this CD4 T cells is also called the helper T cell. Why they are called helper T cell? Because they provide help to both to the humoral immunity, the lower one, along with it also helps to the cytotoxic immunity or cell mediated immunity, that is the upper one. You know that mainly the cytotoxic cell mediated immunity is mediated by this upper one, CD8 T cells. So you know these all B cell, T cell, they are actually they generated from the lymphoid cells, and there is precursor B cell, precursor T cell, then they become immature B cell or T cell, then they progressively mature and they become after that they become active and mediate different immunoresponse. Now another concept is the MHC concept, that is the major, what is the full form of MHC? You know, the major histocompatibility complex. Okay, the MHC molecule, that is of three type, MHC 1, 2 and 3. Now you know the MHC 1 molecule is present in all nucleated cells. Very important concept. All nucleated cells, they contain MHC 1 molecule, and the MHC 2 molecule, that is only present in the antigen presenting cell. Antigen presenting cell. What are the example of antigen presenting cell? You know, macrophage, dendritic cell. Okay, so these are the example of the antigen presenting cell. Now to understand this immunology map, first you have to concentrate on the action of the CD4 T cell, that is the helper T cell, because it is the crucial, it has a crucial role in the mediating both humoral immunity and cytotoxic immunity. If any disease, if it affects the CD4 T cell count or decrease the CD4 T cell count, it will affect both, it will decrease both, or it will hamper both humoral immunity as well as cytotoxic immunity. Okay. Now what are the disease? You know the HIV attacks the CD4 T cells. They multiply inside the CD4 T cells and destroy the CD4 T cell, thereby leading to decrease in number of CD4 T cell. So what happens in the HIV patients? As CD4 T cells degrade, both humoral immunity and cytotoxic immunity both get hampered. Thereby the patient become prone to different type of infection. So my point of discussion is that the CD4 T cell, it has a crucial role. It is the pivot of the all immunoresponse map. The whole immunoresponse map is pivoted by these CD4 T cells, the middle one. Okay, so I am zooming it. Please see, there is stem cells found in the bone marrow, and these stem cells are the precursor of all these lymphoid progenitor cells. See, these are the lymphoid progenitor cells. Okay, and from these lymphoid progenitor cells, there is different type of cells, CD4 T cell, CD8 T cell, B cells, they develops. Okay. Now, and you know the primary lymphoid organs and secondary lymphoid organs. Primary lymphoid organs are found in the bone marrow, and after that they also found in the different stem cells. They are found in the different bone marrow or in the thymus or in the lymph nodes, and after that there is a phenomenon that is called selection in the secondary lymphoid organs that occurs, and after that they release into the main circulation. So this is another topic, please read it from books, but my topic of discussion of today's class is about the immunoresponse. So from these stem cells, their lymphoid progenitor cells, they generated and they get selected in the thymus, and after that selection in the thymus, they release into the peripheral circulation. Okay. So this is the pictorial depiction of the CD4 T cells. The CD4 is the lower one, the sickle shaped structures arising from the T cell. This is the CD4 marker of the CD4 T cells, and the middle one is the CD4 T cell receptors. Okay. So what happens? CD4 T cells, it is the receptor of the CD4 T cell only combines with the antigen that is presented with the MHC2 molecule. Already one mnemonic I have shared, that is the MHC2 bind with the 4, means 2 into 4 is equal to 8, but see the upper one, the CD8 cell bind with the MHC1 molecule. MHC1 molecule is present in all nucleated cells, and that will be 8 into 1 is equal to 8. So total, the multiplication, after the multiplication, total should be 8. 4 into 2, it is just a mnemonic to remember, but it is very important mnemonics. Please remember and understand this. The 2 into 4 is equal to 8. So the MHC2 molecule bind with the CD4 T cells, and 1 is to 8 is equal to 8. 1 means MHC1 molecule bind with the CD8 cells. So CD4 T cells, the middle one, concentrate on the middle one, the middle row. Okay, middle rows, please see the CD4 T cell is coming into the peripheral circulation. Suppose there is any extracellular pathogen entered inside the body. Extracellular pathogen, you know what is the extracellular pathogen? Almost all the bacteria are the extracellular pathogen, except few bacteria like Mycobacterium tuberculosis, lepri, chlamydia, rickettsia, mycoplasma. Rest all the bacteria in the microbiology, they are extracellular pathogen. Okay, and this extracellular pathogen, usually they are engulfed by macrophage or dendritic cells, and these are the antigen presenting cells. So this antigen presenting cell macrophage engulf the bacteria. Okay, and they process inside the macrophage and present these antigenic fragments of the bacteria with the help of the MHC2 molecule. MHC2 molecule is present on the surface of the macrophage or any antigen presenting cells. Okay, so extracellular pathogen, first it is get engulfed by the antigen presenting cell, then they are processed, and this epitope of antigen, epitope of the extracellular pathogen bacterial antigen epitope is presented with the help of the MHC2 molecule to which T cell? CD4 T cells. CD4 T cells. So the CD4 T cell is coming in the picture. Now what is happening? So continuation of the middle row. See the peptide. Peptide means the antigenic epitope of the extracellular bacteria that is presenting with the presented of the MHC 2 molecule of the antigen presenting cell to the CD4 T cell receptor. CD4 T cell receptors. Okay. And you know the CD4 T cell receptor also, it has a co-receptor. What is the name of this co-receptor? See, this is beautifully given by picture. CD28 molecules. CD28 molecules, the upper one is projecting from the CD4 T cells or helper T cells, and it binds with the B cell co-receptor of the antigen presenting cell. So there is main two receptor. Along with that, both antigen presenting cell and the helper T cell both have some co-receptors. The co-receptor of the helper T cell is called the CD28 molecule, and the antigen presenting cell co-receptor is called B7 molecule. So the B7 molecule and the CD28 both combine. Along with that, in the middle, see that the MHC2 molecule is presenting the antigenic epitope, the triangular structures with the antigenic epitope that is presented by the macrophage to the T cell receptors. The T-like structure is called the T cell receptors, and to the TH molecule. TH stands for helper. Okay, H stands for helper. So after this binding, so there are three signals. If you read the Ananthanana Panikkar book or any textbook of microbiology, it has been written that for activation of the helper T cell, there is need of three signals. So what are the three signals? First, see the binding of the MHC2 molecule with the T cell receptor, sandwich in between. Okay, sandwich, the antigenic epitope is sandwiched in between the T cell receptors and the MHC2 molecule, the middle one. The second signal is coming from the co-receptor binding. Co-receptor CD28 molecule bind with the P7 molecule. It gives the second signal. Along with that, after this, what happens? That helper T cell release some cytokine. Cytokine, what is the name of the cytokine? Interleukin 2. So after this first and second signal, what happens? The CD4 T cell, the TH cell get activated and they release some cytokine, interleukin 2, that acts on the TH cell receptor itself. So it has some autocrine effect. Autocrine effect means the source of the cytokine and the source of the cytokine interleukin 2 is the helper T cells, and this interleukin 2 acts on the same cell. So this type of action is called the autocrine effect. Autocrine effect. Okay. So this autocrine effect gives the third signal. Autocrine effect gives the third signal. After that, three signals, when it has been completed, the the helper T cell get activated. They get activated and they divide into two subtypes. One is TH1, the upper 3, see upper 3 one is written as TH1. This one stands for subtype 1, and the lower one is the TH2, that is the subtype 2. Okay. So there are two subtype of helper T cell. It is written in the textbook, but you will understand better if it is given in the pictorial form. That is why I am explaining this picture. So after this division into two groups, TH1 and TH2, what happens? The TH1 release again interleukin 2. Again TH1, the subtype of one of the helper T cell release interleukin 2 that provide helps to the upper row. Upper row means cytotoxic immunity. Okay. and Th2 subtype provide help to the lower row. Lower row means humoral immunity. Okay. When I will discuss the cytotoxic humoral immunity, you will understand how it helps. But for the time being, just you understand the two subtypes, Th1 and Th2. Th1 give helps to the cytotoxic immunity in the form of interleukin 2, and Th2 helps in the B cell immunity or the humoral immunity or antibody immunity. That was the lower one. Please see the Th2 coming to the lower one and it helps to the B cell actually. Okay, so this Th1, beside giving help to the upper row, it also release some tumor necrosis factor alpha and beta, and also it release some interferon gamma. They are all cytokine. Okay, that acts on the macrophage. And this macrophage get activated. They release some enzyme, lysozymes, and they kill the bacteria, extracellular bacteria, killing of intravesicular pathogen. Right? You know what is the function of macrophage? They engulf. Then there is a phagolysosome formation, and with the help of lysosomal enzyme, they degrade the intracellular bacteria. So this beside providing help to the cytotoxic immunity and humoral immunity, the CD4 T cell also helps in the activation of the antigen presenting cell itself. Antigen presenting cell already engulfed the bacteria, so the bacteria now is intracellular, means inside the macrophage. So if this macrophage itself get activated by this tumor necrosis factor alpha, beta or gamma, what happens? They form the phagolysosome, and with the help of lysogenic enzyme, they can degrade the intracellular bacteria. Okay. Beside that, this Th1 also release interleukin 2, interferon gamma, that activates the natural killer cell. You know there is another type of natural cell that is present in the innate immunity, that is called the natural killer cell. When they get activated, they can also kill the intracellular bacteria. Okay, so this is the story of CD4 T cells. So starting from thymus, after getting selected by the thymus, the CD4 T cells release into the peripheral circulation. Then what happens? When any bacteria, extracellular pathogen enter inside the body, they get engulfed by the macrophage and they presented with the MHC2 molecule to these CD4 T cells. There is three signaling. One is this binding of the MHC2 molecule with the T cell receptor. Second one, the co-receptor binding B7 of the macrophage or antigen presenting cell, and the CD20 of the helper T cells, and the interleukin 2 that is liberated and acted upon the Th cell itself. Okay, so three signals. After these three signals, what happens? They divide into two subtypes, Th1 and Th2. Th1 helps to the cytotoxic immunity in the form of interleukin 2, and Th2 participate in the humoral immunity and helps into the humoral immunity. Beside that, this helper T cell release some cytokine that activates the macrophage and natural killer cell that kill the bacteria. Okay. Beside that, some Th1 becomes the memory T cells. What is the function of memory T cells? If suppose there was any bacteria enter inside the body and stimulate this subtype 1 of the CD4 T cells, it takes a memory. It store a memory of this pathogen. Okay, and subsequently, if there is a infection by the same bacteria, what happens? This action become very much quick. That is called the secondary immunoresponse. Secondary, the primary is the first time, and the secondary immunization occur when the same bacteria enter inside the body second time. And the secondary immunoresponse is very much fast. Why it is fast? Because of presence of these memory T cells. Memory T cells, if present inside the body to a specific bacteria, what happens? This action, this activation of the macrophage, activation of the natural killer cell, activation of the cytotoxic and humoral immunity occurs very much fast. Okay, so that is, and it is due to this fast action is due to these memory T cells. Okay, so this is the story of CD4 T cells or helper T cells. Okay, that has a pivotal role. Why pivotal role or central role? Because it is giving help to the both humoral as well as cytotoxic immunity. Both arm of immunoresponse is getting help from the CD4 T cells. Okay. Now I will discuss about the upper row. Upper row means it is the cytotoxic immunity. Cytotoxic immunity is mediated by which cell? CD8 T cells. CD8 cluster of differentiation type 8. So this CD8 cell, and it is a lymphocyte, it is originated from the lymphoid progenitor cells, then they get selected in the thymus, okay, and after this selection they enter into the circulation. Okay, now suppose the pathogen or the foreign body is intracellular. You know, all the virus are not extracellular, all virus are intracellular pathogen. So, and some bacteria like Mycobacterium tuberculosis, Mycobacterium leprae, Mycoplasma, Chlamydia, Rickettsia, they are also intracellular. Fungus, parasite, they are also intracellular. So, the extracellular pathogen usually killed by this B cell immunity or antibody mediated immunity. Okay. But the intracellular pathogen like virus, fungus, parasites and Mycobacterium tuberculosis, leprae, they are killed by cytotoxic immunity, because these pathogen or these foreign body, they are intracellular. Okay. So virus can affect any cell of our body, and you know any cell of our body is nucleated cells. Because all cells are nucleated except RBC. RBCs are not non-nucleated. Besides this RBC, all cells of our body are nucleated cells. Okay. And virus, fungus, pathogen, virus, fungus, parasite, they can infect any nucleated cells of our body. And you already know any nucleated cell express which MHC molecule? MHC1 molecule. MHC1 molecule. Okay. So suppose one cell, it may be neuron, it may be central nervous system cell, it may be the lymphoid cell, it may be the skin tissue cell, it may be the gastrointestinal cells. They are infected by some virus or fungus or parasite. So what happens? See, this is the picture of the upper row. This is a virus infected dendritic cells. Dendritic cell is a central nervous system cells. Okay. Now this virus is engulfed, virus is destroyed and divided into different particles like viral peptides, and this viral peptide is an antigenic epitope. It is presented with the MHC1 molecule. To which T cell? CD8 T cell. Why? 1 into 8 is equal to 8. Okay, this is the formula to remember. Okay, so MHC1 molecule is present in all nucleated cells. So when any virus or fungus or parasite is attacking any cells, these cells are nucleated cells, and these nucleated cells express MHC1 molecule. So with the help of this MHC1 molecule, this cell express outside of the cell to the CD8 T cells or cytotoxic T cell. This one is the cytotoxic T cells. Okay. Now continue on the right side, same row at the upper row. What is happening? Again there is a story of co-receptor. Again the cytotoxic T cells also express some co-receptor, that is called the CD28, and these any cell of our body, nucleated cell, they also express the B7 co-receptors. So there is a binding of the T cell receptor along with the MHC1 molecule, and in between there is the viral peptides is presented to the T cell receptor. Please see the triangular structure. This is the viral peptide. It is sandwiched between the T cell receptors and the MHC1 molecule, okay, of the antigen presenting cells. This is the first signal. Second signal, again the co-receptor binding. See, CD828 is binding with the B7 molecule. It gives a signal. And third signal is coming from Th1 subtype of helper T cell. That is why the CD4 T cell is providing help to the cytotoxic T cells in the form of interleukin 2. So it acts as a third signal for activation of the cytotoxic T cell. After these three signals, what happens? The cytotoxic T cell become mature cytotoxic T cell and get activated. After get these three signals, these immature cytotoxic T cell become mature cytotoxic T cells, and what they do, with the help of two enzymes, one is the perforin, another one is the granzyme. You know what is perforin? Perforin is released by cytotoxic T cell. What they do, they produce pore surrounding the virus infected cells, and they enter some enzyme that is called the granzyme. With the help of granzymes, these viral infected cell, they start to be apoptotized, means the apoptosis of the virus infected cell starts. So in this way, the cytotoxic T cells help in the killing of the virus. It cannot directly kill the virus, but it can kill the viral infected cells, virus infected cells. Besides that, similar to the helper T cell, the cytotoxic T cell, some cytotoxic T cell become the memory cytotoxic T cells, thereby leading to the fast action of the cytotoxic T cells in the secondary infection or secondary immunoresponse. So same concept. Okay, the primary immunoresponse is a little bit delayed, but the secondary immunoresponse is very much fast due to presence of these memory T cell and memory cytotoxic T cell and helper T cells. Now this is the story of the cytotoxic immunity. Now coming to the lower one, the lower row, that is the story of humoral immunity. So humoral immunity, in case of humoral immunity, what is happening? It is also killing the extracellular bacteria or extracellular pathogen, not the virus. But in that case, there is the antigen. Antigen is presented by the B cell itself. In last upper two case, upper row, please see the antigen is presenting with the either antigen presenting cells or virus infected dendritic cell or the any body cells. But please see in the lower row, the B cell have some receptors for the antigen, extracellular antigen or extracellular foreign body. It is usually the IgM receptor or IgM that is present on the surface of the B cell that acts as a receptor for the antigen. So this antigen is binding with the IgM on the surface of the B cells. Then they get internalized. The antigen is internalized. Then they are processed into small fragments, and this small fragment as a peptide is presented by the B cell. So the B cell, now it is acting as an antigen presenting cells. Okay, this epitope is presented to the which cell? Th2 subtype of the helper T cell. Subtype 2 of the helper T cell. That is why again the helper T cell is providing help to this lower row also. Means it is giving help to the humoral immunity also in this way. Okay, so the B cell is presenting the peptide or the foreign antigen peptide to the receptor, T cell receptor of the subtype 2 of the helper T cells. Please see this one. And after this, it acts as a fast signal, and after these signals there is no co-receptor binding in that case. But after this fast signal, what happens? The Th2 subtype of cell, they get activated and they release some cytokine. But this cytokine is not interleukin 2, it is interleukin 4, 5 and 6. Please remember this, very important concept. So after binding of this B cell receptor, after presenting this B cell is presenting the peptide to the T cell receptor. Which T cell? Helper T cell. Which subtype? Subtype 2. What happens? The Th2 cell get activated and they release some cytokine. Which cytokine? Interleukin 4, 5 and 6. It acts on which cell? B cell. This Th2 cell is releasing interleukin 4, 5, 6. That activates the B cell. And transfer the B cell to the plasma cell. What is the function of plasma cell? You know, plasma cell produce antibody. So the antibody producing B cell is called the plasma cell. And B cell only produce antibody, only becomes plasma cell after this getting stimulated by this interleukin 4, 5, 6. And this interleukin 4, 5, 6 is secreted from which cell? It is from the helper T cell. That is the helper T cell. That is why I am telling again and again why they are called helper, because they are helping to the upper row, that is a cytotoxic T cell in the form of interleukin 2, plus as well as they are giving help to the B cell also in the form of interleukin 4, 5, 6. So this is the main concept you have to understand. Okay. That is why they are called helper T cell. And if this helper T cell is destroyed in any condition like HIV, the both arm of the immunoresponse get destroyed. Okay, thereby leading to different type of infection. Okay, so this plasma cell produce antibody, and this, what is the function of antibody? You know, they can neutralize the extracellular toxin. They can opsonize the bacteria. What is opsonization? It is the process by which the bacteria becomes tasty to the macrophage. So when this opsonization means they produce some substance, the antibody when binds with the bacteria, the bacteria become very much tasty or very much attractive to the macrophage. Okay, and macrophage easily they can engulf this opsonized bacteria, and this opsonization occur by the help of this antibody. When this bacteria is coated by antibody, they become easily identified and engulfed by the macrophage. That this process is called opsonization. Opsonization meaning that means that the process by which the bacteria become tasty or attractive to the macrophage. Okay, so macrophage easily engulf them and they can kill them. Okay. Beside that, this antibody, they activate the complement. You know, there is two pathway of complement. One is the classical pathway and another one is the alternate pathway. Okay, and this antibody IgM specifically, they helps into the complement activation, and the antibody sometimes they can help into the neutralization of the virus also. Okay, so this is the function of the humoral immunity. But how this humoral immunity get activated? With the help of CD4 T cells. Why? The CD4 T cell is releasing interleukin 4, 5, 6, that is activating the B cell. After this activation, B cell is transforming to the plasma cell, and you know the only plasma cell, not the B cell, only the plasma cell can produce antibody, that is the humoral immunity. Okay. So this is the immunology, whole immunology map. Please understand this. Okay, listen it again and again, and please see this paper. This, take a printout from this WhatsApp group and stick it on your wall, reading room wall, and please see it regularly on daily basis to understand this concept, so that you can upload or you can register this picture in your brain for your lifetime. And I promise that if you do this and if you understand this paper, this will be helpful for not only for microbiology but also for the medicine and for your whole medical career. Okay, so please understand this. You will not get this paper or this picture in any textbook or any even the Robin's pathology which is given, that is little bit complicated, not so much easy to understand. The Robin's pathology that is given immunoresponse. So please take or utilize this opportunity and understand this, and definitely it will help to understand the different difficult concept of the immunology as well as the different concept of the medicine also. So thank you for listening, and hopefully it will be helpful for you. If there is any query, please ask in the WhatsApp group, and I will try to answer it as soon as possible. So happy Diwali in advance, and this is a little gift for you for Diwali, and you will understand. I think that you will understand it will be helpful for your medical career for